In this video, I'm going to be talking about nucleotides, DNA. So this right here is a nucleotide, nucleotide. I'm going to be talking about nucleotides, DNA, and I am going to get a little bit into transcription and translation. So transcription and translation, which is known as the central dogma of biology. That central dogma. So the central dogma is that we go from DNA, it's transcribed into RNA, and that's translated into proteins. All right, so we'll start here at DNA then. So DNA is composed of these nucleotides, and so this shows the nucleotide. It has the phosphate singled out on it, and has the ribose. It's a deoxyribose. We can see over here, deoxyribose has just hydrogens on this 2' carbon, and ribose has a hydroxyl group here. So deoxy, no oxy, no oxygen. It's deoxyribose, or 2-deoxyribose, because it is the 2' carbon. And I'm calling it 2' because in these nucleotides, it's the base here that has the non-primed numbers, and so the sugar gets primed numbers just to differentiate the numbering from the base. And so this shows the base with an adenine on it. It can also be any one of these other ones over here. These are sort of the frameworks of the two types of base called purines and pyrimidines. So purines have these double rings on the base. So adenine is differentiated. It has this amino group up here. Guanine has a double bond oxygen with an amino group here, and in fact, this amino group here, along with these other two nitrogens, is called a guanidium group. So guanidium, I think that's how it's spelled. So that's where the guanine comes from. So this 3 nitrogen sort of motif here, or moiety, with all bonded to this central carbon, that's called a guanidium. And then we have the pyrimidines, which are all this single ring structure here, and so cytosine has this primary amine up here. Thymine has this double bonded oxygen rather than this primary amine, and then thymine and uracil are the same, except uracil is found in RNA and thymine is found in DNA. So uracil will be bound to sugars like this that have the hydroxyl group on it, DNA to these deoxyribose. And the difference between them is just this methyl group here. So that's the only difference between thymine and uracil. That's just sort of a way for cells to tell the difference between DNA and RNA. And so, yeah, though that's basically all there is to well, it's not all there is to it. We're going to get into more, but well, moving on. So this, like I said, is a nucleotide. It has this base bound to it. It has a phosphate bound to it. Uh, and in fact, it can have more than one phosphate bound to it. And that's when you get uh, ATP. So if you've taken like a biology class, you've probably heard of ATP. That's energy, and the energy comes from these phosphodiester bonds. So if you remember from the chemistry introduction, I talked about an ester, which was a carbon to a carbon double bond, oxygen to an oxygen to a carbon, and that can go on to whatever else. So this was an ester bond. And so here, instead of the carbon double bond, it's a phosphorus double bond to an oxygen and then to another phosphorus double bond. So that's called a a phosphodiester. And it's diester because we see that it's on both sides of this oxygen here. And so these phosphodiester bonds contain energy because to produce this molecule, to add these phosphates onto it, is actually, if we have our energy diagram here, so energy in the reaction, it, we're actually at a lower energy when we don't have these phosphates on it. So the ATP would be up here. If it just has a single phosphate on it, like we show up here, then it's called adenosine monophosphate, which is also AMP. So adding those two phosphates onto it make it into ATP. You can also add one on, and then it would be adenosine diphosphate, which would be ADP. So we can have AMP, ADP, and ATP. And there's energy in these phosphodiester bonds that when we go backwards in this reaction from ATP to AMP, we are releasing that amount of energy right there. And so that's how we sort of use this ATP, and I'm specifically talking about ATP because that's the one that's primarily used for energy. There is sometimes used GTP, which then is the same thing, except instead of having this adenine on it, we would have a guanine on it. We'll run into that once we get into metabolism. But for the most part, ATP, I think I described it in an earlier video as sort of the $20 bill of energy in the human body, because this is kind of the one that's used the most often, sort of like $20 bills. And so other molecules that we will run across that are made out of these nucleotides. So this one is NAD, or yeah, NAD, should be a D plus, which stands for nicotinamide adenine dinucleotide. And so down here we have this nicotinamide group on it. And you can see I have nicotinamide by itself up here. So we have that on here. And we have this adenine up here. And so this right here would be our AMP, or adenosine monophosphate. Then down here we have a nicotinamide as our base. And I sort of suggestively wrote or put this here, this niacin, or vitamin B3. You can see that this is very similar to nicotinamide, which is just different by having this instead of this. And so I've suggestively drawn that here because we need niacin, vitamin B3, in order to produce this. This is another form of energy carrier that we have, but instead of carrying the energy as these phosphodiester bonds, it's actually carried in the form of a redox reaction. So we see here that this is our nicotinamide right here, and we see that this nitrogen right here is having a hydrogen and 2 electrons attached to it, and so that neutralizes it and turns it into NADH. So no plus because we don't have a charge, and H because we have added hydrogens onto this, and we see that this double bond here. So one of the hydrogens is being added here, which pushes that double bond there, then pushes those electrons there. And that's why we end up with the double bonds here with this extra hydrogen down here and no charge on our nitrogen. And so carrying these electrons around is the way that NADH carries energy, and we'll see how that is used as energy once we get further along into metabolism. So the electrons are used to produce energy by having it move down a potential gradient, and oxygen is actually the final acceptor of it. That's why we need oxygen. So NADH will drop off its electrons, turning it back into NAD plus, and that let those electrons go down. So there's a bunch of different proteins in this process here. The electrons are being passed from protein to protein, but oxygen, since it has such a high electronegativity, is the final receptor of those electrons, which turn it into H2O. There's also hydrogens out there. So to balance it, we would do that. Yeah, so we'll go through this whole process later on, or in a different video, in a later video, when we talk about this process. I mean, this is still just turning AMP into ATP, so having this electron move down this gradient is turning AMP into ATP. And so these NADH molecules are still just kind of a way of getting to ATP, which, like I said, is our $20 bill here as far as energy goes. All right, so let's move on to the actual structure of DNA. And DNA has a structure that's well conserved, and that's because of the way that these base pairs pair up. And so we always have oops, that doesn't look very good. We always have A pairing with T, so adenine and thymine, or in RNA it would be uracil, but RNA doesn't really form this double helix, and G always pairs with C. So we can see that up here. We have our A and our T, and we see that they come together with these hydrogen bonds. And then we have our G and our C, which come together with hydrogen bonds. And one thing you'll notice is the hydrogen bonds on here, there's only 2, and there's 3 on here. So what that means is in parts of the DNA strand where you have lots of GC content, then our DNA is actually held together stronger. Whereas if you have A's and T's, a lot of A's and T's, then it's not held together as strong, and the DNA double helix can come apart more easily. We would say that its melting temperature, its melting temp, is affected by these AT and these CG content of the DNA. And so if we have high GC, then melting temperature will be up. If we have high AT, melting temperature will be lower. It'll be easier to separate these strands. And so if we have mispaired, if we had, let's just say an A and a C, and so that's a purine with a pyrimidine still, the hydrogen bonds would not match up, and so this would not is less likely to happen, and it creates sort of a deformation in the actual DNA strand. So we see over here, the DNA strand has a very regular structure. So it's 20 angstroms, 2 nanometers across, and one helical turn is 34 angstroms, which is 10.5 of these base pairs. So we have a turn every 10.5 base pairs. You should also notice that DNA has a right-handed turn. So if we hold our right hand with our thumb up and then curl our fingers in like this, as we move up the DNA, then the DNA is twisting in the direction of our fingers. So you'll see a lot of times in movies and TV shows, they will show DNA with a left-handed turn. So now that you are cursed with this knowledge, whenever you see that, you can tell your friends that that's fake. So anyway, DNA has this nice, really regular structure, and so if you get this mismatched base pairing, it will deform this structure, which your cells have proteins that can move along this, and they will find those deformations, and they can sort of fix them. They can take out if you have a AC, it might come along and cut this off and replace it with a T, the way it's supposed to be. And this would also work with other pyrimidine purine mismatches, but you can see also, if we go back to, well, I guess we can see it here, our purines are quite a bit bigger because they have these double rings, and our pyrimidines are smaller. So if we had 2 purines, if we had A to G, then we can see that this would cause this to bulge out quite a bit. Whereas if we had pyrimidine to a pyrimidine, then we could see that it would sort of give it sort of an indentation. And so those are also deformations that our DNA proofreading proteins can find and fix that, because we don't want to have those mutations in there. So yeah, like I said, our DNA has this nice structure. You should also notice that it does have 2 different size grooves. Their DNA does not have like a nice perfect sort of, this isn't perfect, my handwriting is terrible, but you can see that there are different size grooves on here, and they're called the minor groove and the major groove, which as you can see, the minor groove is 12 angstroms and the major groove is 22. And that's important because proteins that attach to our DNA when they attach to a specific site in our DNA, they are sort of probing their protein fingers into these major grooves and looking at the base pairing going on in the major groove. Um, and so I'll show you later that there's sort of a code in the hydrogen bonding pattern of our base pairs that can be read from the major groove, and that's what those proteins will be looking for. So down here we'll look at the difference between DNA and RNA. So the first thing to notice is that DNA is most of the time, almost always double-stranded. RNA is kind of a wild card. The sort of standard view is that RNA is single-stranded, and even this is a bit simplified because most of the time RNA will sort of fold up on itself and create these sort of weird structures. So they'll have base pairing to themselves going on here, going on here, going on here, and they'll have these sort of loops at the ends. But the way we sort of just think about it is as this single strand like this. And we talked about before that that RNA has this uracil and DNA has this thymine, and we talked about how DNA has just a hydrogen on the 2' carbon, while RNA has an oxygen. And what you end up seeing is it doesn't show it here, but on RNA, so this should have a hydroxyl on it to make it the ribose that you would use on RNA. And so the sugar, the ribose on RNA has this it's called a C3' endo pucker. So this is called a pucker. Oops, pucker. That almost looks like a swear word. Pucker. Okay, and so RNA has this 3' endo pucker, and DNA, which, like I said, doesn't have the hydroxyl here, has the 2' endo pucker, and you can see that this says B conformation here, and what we will see is that DNA is usually in this B conformation here. And so we see there's this A conformation which has a very different geometry. You can see that these things are highly angled, and we have different sizes of our grooves here. B DNA, the B conformation, you can see looking down through it too that it has this sort of conformation. bDNA has the base pairing going on in here, and it has this nice regular structure. zDNA, which is interesting too because this does actually have a left-handed helix. And so this isn't found very often. You can generate this with a very specific solution of different salt concentrations. I don't really remember how you go about forming aDNA, but for our purposes, we're just going to think about bDNA. And so bDNA has that structure that we talked about above. We can look at how the base pairs can have these different orientations to each other. I'm not going to go through this whole chart, but you can see that that there are different sorts of offsets that they can have. They can be at different angles, and so bDNA and aDNA have sort of canonical offsets and angles for the base pair parameters and the base pair step parameters and the local helical parameters. And so the different angles and offsets and everything are pretty well characterized in DNA, and these are the ones here that occur in our bDNA, which is what we are interested in. I mean, you don't have to memorize this. It's probably almost never going to come up again, but it's. I'm just pointing out that that there are these different geometries that we can find in our DNA, and the DNA that we all know and love have these offsets and these tilts and so forth, rise, slide, twist, those kind of things. All right, so DNA, once we have it in our double helical strand, then what happens is DNA, so we have these 8 proteins, these octamers, which are, these are called, so these are the histones here, the histones, and they create these things called nucleosomes. Nucleosome. And so these proteins, there's 8 different proteins. The DNA wraps around it, and then we can start creating our sort of higher order structures of DNA. I show here these structures of the nucleosomes. So this sort of lighter blue stuff here is the DNA that's wrapped around it, and the different colors here are the different subunits of the histones. And we can see here, so this, it shows it as negative, but this is potential. And so negative potential is telling us that we are going to have some attraction going on here. This is showing just the DNA part of the nucleosome, and so we see it makes this turn. You can actually see here at the side view, it makes these 2 turns around it, and we see that it makes a turn that is a left-handed turn around the histone. And so if we had our hand like this, if we had our thumb on our left hand right here, and then we had our fingers, then as we go this way, then we're curling around in the left-handed turn. So the DNA double helix is right-handed. The way DNA is wrapped around these histones is left-handed. And we can see down here just another structure of the nucleosomes. Up here we have the nucleosomes and the different subunits of our histones. We have the sequence here, and you can see that there is a lot of arginines in it, and arginine actually has well, I have it down here actually. I don't need to draw it. It has this guanidium group on it. So at physiological pH, this would actually have another hydrogen bound onto this nitrogen, and it would be a plus charge. And so the plus charge, so this just is a guanidium. We can imagine that we go off to our arginine, and so this is a positive charge in our DNA backbone. So right here is one of our phosphates, and phosphates are negatively charged. And so the arginine, the guanidium moiety at the end of the arginine side chain is forming an ionic bond with the phosphates on the backbone of the DNA. So this is on the backbone. We can see this is the ribose here, the deoxyribose. This is our phosphate. This would be our base here. And so you can see that all these arginines here are going to be forming these salt bridges with the oppositely charged backbone of the DNA, and that is how the DNA is held onto uh, the histones to generate the nucleosomes. All right, so I'm just going to briefly go over some of this stuff. And so this is DNA replication. Um, and so what we see here is this is our DNA coming in. So this is the DNA that is on this side not yet replicated. And since DNA has a directionality, uh, actually, let's go take a look at that picture. I forgot to mention it when we were looking at it. So if we look up here, we see that DNA, each strand has a directionality. This is 5 prime, this is 3 prime. And that has to do with the sugar numbering. And so we see that up here we have our 5 prime carbon, and then we go down to here to our 3 prime carbon. And so we're always going 5 prime to 3 prime. And then this one is antiparallel, so it's 5 prime going up to 3 prime. So we see we have our 5 prime here, we go to 3 prime, and that just repeats over and over. So we have this directionality that's anti-parallel in our DNA double strand. And so when we're replicating DNA, that poses a challenge in that we're separating this DNA strand, and one of them will be in the right direction while the other will be in the wrong direction. And so the right direction one is pretty straightforward. We just send it through this DNA polymerase. It looks at the base pairs on the single strand and then adds the complementary base pairs onto it, onto the strand that it's generating, so that we end up with 2 different strands. But on this side, which is called the lagging strand, we have to sort of circle it around, and then we have to have it kind of going this way along here, and then this will detach from here and then attach further down, further up over there, and so we have this side sort of constantly switching. Actually, I had and there's a link to a video of it in the in the description, but I have this here, which is a GIF of the video. And so you see what's going on here. Our leading strand is just continuously being replicated, whereas on this side we have our lagging strand. We have to keep doing these detaching and retaching. And if you're interested in this, I definitely recommend checking out the video of it in the description. The video has narration and actually talks about what's going on here as this is moving along, and so it describes actually what's going on in pretty good detail. It's a really nice video, and so I definitely recommend checking that out. And so what happens when our nucleotides are coming in? So these DNA polymerases, so there are nucleotides coming in to be attached to the single strand. What happens is we have our single strand, and I'll just draw it like this. So we have a T and a C, and this will just kind of keep on going in both directions. But what happens is when this is inside the DNA polymerase, the other nucleotides, just free nucleotides in the triphosphate form, and so we will have like a GTP, a guanosine triphosphate, and that can come down, and when it senses that that it's pairing with this cytosine here, then when it can form that base pair, it will allow that to base pair, and then it will attach uh, this uh, this guanosine triphosphate onto the strand. So back here, since we've so maybe we could look at it like this. This T has just been added on, and so we have base pairing going on here. And we want to attach this G onto the end right here. And so what we end up having to do there is use the energy in the GTP, because when it's attached on here, it will be in the GMP form. And so once we have that CG, this will be in the GMP form, and so we have what's called a pyrophosphate come off of it, and the release of that pyrophosphate makes it an energetically favorable reaction. And so that's how we can have these that's how we can have all the bases. We'll have our GTP and ATP, as we talked about before, but we'll also have the cytosine triphosphate and the thymine triphosphate. And those are the forms that we have it when it's out in solution. And then these are what come and get attached to our strand as we're adding DNA onto it. So we can also see that if we have something, say we have a CTP here, and that in our DNA polymerase is now around this one, and this comes, it fails to form base pairing here, and so the DNA polymerase will just sort of eject it back out and wait for the correct base to come in here and base pair with this thymine, which would be an adenine. And so I show here the reaction that's happening. And so so this in DNA should actually be just an H. This shouldn't be a hydroxyl in DNA. But this on our 2' carbon, this hydroxyl here comes and attacks this phosphate. And so what we end up with in our active site is something that will look like this. And so we have our 2' carbon here with its hydroxyl group, and that's trying to form a bond with this phosphate here. And the negative charges on all these phosphates is stabilized by these magnesiums. So that's foreshadowing to when we talk about micronutrients and things like that. Magnesium, one of the things it's needed for is as a cofactor in in these DNA polymerases and RNA polymerases, which is what this one would be, because we have we don't have a deoxyribose here, and so this would actually be in an RNA polymerase, but the reaction is pretty much the same, and so we're stabilized by these magnesiums. This is forming a bond with the phosphate here, and then these electrons go on to this here, and then we end up with our pyrophosphate, which will look like something like this. Oops, that should be single bond. We could put negative charges on here. We could put hydrogens. It's not that big of a deal either way, but this is pyrophosphate like this. And actually removing the pyrophosphate rather than just a single phosphate is more energetically favorable. So this is more energetically favorable. So we are actually using quite a bit of energy to add a new base onto our extending chain here, and that's because, like I said, if we look at our energy diagram here, so reaction energy, uh, our our ATP would be up here perhaps, and our ADP would maybe be here, where down here is our AMP. And you see, then if we wanted to go from ADP to ATP, then we'd have to go, oops, draw that better. We'd have to go up through that to ATP. We'd have to go through the same activation energy, even from ADP to ATP. And so you can go from AMP to ATP. Most of the time it's going to go through this right here. But anyway, the point is ATP going back to AMP is more energetic than ATP going down to ADP. And so this is DNA replication is a very energy intensive reaction. It's a very energy intensive process in our cell. Over here, I'm just showing that there's actually there was another way that you could have this replication going on, where pieces of the DNA are just sort of getting stuck wherever. But this semi-conservative and this conservative model were two sort of competing models back in the day when DNA was first being sort of discovered as the genetic material. And it turned out that we go through this semi-conservative model. So what happens is on this DNA double helix, both strands sort of come apart, which is what we see up here. And so you can see just before entering our replication complex here that we have these single strands, and then on each single strand we are adding another strand. And so one strand from, or each of the replicated DNA strands get, or DNA double helices get one strand from the parental strand, rather than having this sort of come apart, replicated DNA, and then this comes back together as its own strand, and the other strand makes its own. And so you would end up with the parental strand staying together. So this model is not what happens. The semi-conservative model is the one that happens to be the case. All right, now I'm going to talk a bit about transcription. I'm not going to go into too much detail, but I do want to just show that. Before we were talking about DNA replication, which is DNA going to make more DNA. So transcription, so this was replication. Right, and now we are going to talk about transcription. Transcription, which is DNA making RNA. And so they call it transcription because when you're transcribing a book, you are just copying word for word, and DNA and RNA are both in the language of nucleotides. So RNA, when you transcribe RNA off of DNA, it's going to have a reversed and opposite order of the nucleotides. But we call it transcription because they're both in the language of nucleotides. In a few minutes, when I talk about translation, that is RNA to protein, and so that's called translation because we're going from the language of nucleotides to the language of amino acids. But here we'll talk about transcription first. So transcription starts at what's called a promoter. So the gene would be all this stuff down here. This promoter is where the transcription starts. So we have this thing, which is called a TATA box, TATA. It's not always exactly TATA, but it's always a thymine and adenine rich area, which, as I said before, makes it easier to split the two strands of the DNA apart here. And so we could have something like this transcription factor 2D come in, and it finds this TATA box, and it can bind onto the DNA there and start separating the two strands. Well, TF2D has a bunch of other functionalities, or even hypothetical functionalities. It's not even it's not fully understood. And in fact, TF2D, the structure of it is actually touching two different parts of the DNA, so it has this sort of arc here. It's a very large protein. But anyway, then these other two, this TF2A and TF2B, are recruited to the promoter on the gene. And if I remember correctly, I think TF2B is the protein that starts sort of digging its fingers in between the two strands of the DNA and starts separating them. I might be wrong on that, but I think TF2B is the one that does that. Then we can have TF2F along with the RNA polymerase. So the RNA polymerase is similar to the DNA polymerase we talked about in replication, except instead of reading the DNA and adding and making another strand of DNA, it will read the DNA and make a strand of RNA, hence RNA polymerase. Polymer being that it's DNA and RNA are long polymers, so it's polymerizing them. And maybe now is a time, better late than never, to say when you see ASE at the end of the name of a protein like that, that means it is an enzyme. And so the RNA polymerase and the DNA polymerase are both enzymes because they catalyze reactions. And then we recruit the TF2H and the TF2E. TF2H is another protein that's still being studied quite thoroughly. It has a lot of different functionalities that are not completely 100% understood. And so we recruit the TF2E and TF2H. And then we have what's here called the pre-initiation complex. And so we have all our transcription factors recruited. And then what happens is the TF2H, which has it has an ATP, or it can use ATP to actually phosphorylate the RNA polymerase. And once the RNA polymerase is phosphorylated enough, because it undergoes multiple phosphorylations on what's called its C-terminal domain. So one of the subunits, RNA polymerase, is actually composed of numerous subunits. Forget exactly how many. It might even be somewhere in the range of 12. But one of them has this tail coming off of it. The protein has this sort of floppy part coming off of it that has these repeated amino acids, and so it contains lots of serines and actually I don't know if it has threonines. I don't remember exactly. It has serines though, and so that can be phosphorylated at these different sites. And so we get multiple phosphorylations on the C-terminal domain, and that is what sort of signals for transcription to begin. Um, so transcription beginning, well, there's a whole process to that too, because this RNA polymerase can actually pause partially into the transcription, and the lab that I worked in my postgraduate work, there were people trying to figure out what that pausing was and what its function is, and how it actually happens, what's causing it to pause, and that's still an area of active research in the lab that I worked in, as well as other places. Um, Up here, I just have these actual structures of the transcription factors. So we have the so TBP is part of that TF2D. The TF2D is a multi-subunit protein in itself, and one of the proteins in it is the TBP, which is called the TATA binding protein. So it's looking for this TATA box, and it will bind to it. And then we can add in the other proteins, and we end up getting our pre-initiation complex. Up here, I wanted to show that when in gene expression, so there are these enhancer regions that are upstream. So our DNA is in this direction, or at least the strand where our gene of interest is going in this direction. So this is upstream, and it can be quite a ways upstream. It can be thousands of base pairs upstream. And so proteins called transcription factors, you'll also often see as TF. That's what these are. These ones are called the general transcription factors because these are involved in all transcription. But these transcription factors, these that bind to enhancer regions, these respond to cell signaling. And so a cell signal you might be familiar with would be something like insulin. And so insulin binds to the outside of the cell. There's a signal transduction, which signal transductions we'll go into more in later videos, but the tail end of that signal transduction is that this transcription factor is modified in some way, usually by phosphorylation, and that will allow it to come in and bind to this enhancer. Then what happens is this giant protein complex here called mediator can bind to the transcription factors at the enhancers, and then mediator, true to its name, will actually mediate the signal between these transcription factors and transcription. So mediator sort of recruits the general transcription factor machinery to come down and bind onto the promoter and generate this pre-initiation complex, which can then begin to elongate, can go into elongation or transcription. We see that there are activators, there are enhancers that can be very close. These proximal enhancers can be very close to the promoter of the gene. And in fact, in things like yeast, all of the enhancers are very close to the gene. It's only in well, I guess you could call them higher animals if you want. Things like mammals and reptiles and so forth can have these enhancer sites much further upstream. And so mediator is the protein that can well, mediate between the enhancers. And when there's transcription factors bound to these enhancers, mediator finds it and says, oh, I guess this gene needs to be turned on, and it will recruit the transcription to the promoter, and then that can start transcribing. Uh, you'll see here that we have our nucleosomes on the DNA. There are various, not completely understood ways that the RNA polymerase can sort of move around these and keep continuing on. And in fact, transcription can be regulated by via modifications to these nucleosomes. So if you've heard of things like epigenetics, it's on these genetics. It's on these nucleosomes where epigenetic control of gene expression happens. I'm not going to go too deep into that. That's a whole area of research. In fact, another professor at the university I did my postgraduate work is working quite a bit on that. And in fact, she is the one back in the 90s who first got the structure of the nucleosome. Carolyn Luger. But anyway, so yeah, that's how gene expression works. Like I said, we'll go through this signal transduction steps. There's a lot of different ways that can happen before you get from your signal molecule to the transcription factor, which then comes to the enhancer. And yeah, I'm not going to repeat myself. So when the RNA comes off of here, so you can see, so when you're transcribing, you would start having an RNA strand coming out of here that's growing as this moves down the DNA, because you're doing this reaction down here, adding new nucleotides onto the RNA. So the RNA that comes off can be in one of a few forms. The one that's sort of the canonical form is what's called mRNA for messenger RNA. And the sequence on that will actually have only this part of it will be the protein coding sequence. But then it also has this 5' untranslated region and this 3' untranslated region, and different proteins can bind these regions, and that will regulate the way that the RNA is sort of sequestered in the cell, or the way that it's translated and things like that. We also have this 5' end cap, and what that is it's this methylated guanosine here, and that's attached in this sort of weird way, because it's on the 5th carbon here going to this 5th carbon here, so it's not in the usual 3' to 5', and we have this triphosphate here. And this just protects the front end of it from being degraded. So we don't want our RNA to be degraded, so that the cell puts this 5' cap onto it. And we also have this poly A tail, so that's just a bunch of adenines added to the 3' end of our mRNA. And here I just have a lot of the different ways that that RNA is regulated. So once you have the RNA, it doesn't just sort of immediately go to translation. There are a number of different things that can happen. So in the nucleus, we have this thing called alternative splicing. So in the gene, there are actually only these areas called exons that are transcribed. Then there's these places in between called intron. So intron that are not transcribed, but you can differentially bind these exons together. So you could have you could transcribe just exon 1 and exon 2 to make an mRNA, or exon 1 and exon 3 skipping over exon 2 to make a different mRNA, and exon 2 to exon 3, and there can be many more exons going on down the gene. So there's this alternative splicing that can happen, that can change the mRNA that you get from the very same gene. So then the mRNA can be exported out into the cytoplasm where the ribosomes are, which is where translation happens, and I'll get to that in just a little bit. And so if we have our sort of mRNA here that's ready to go, you see this has some proteins on the 5' UTR and 3' UTR. So the one thing we can obviously do is translate it and turn it into a well, we don't turn it into, we translate it into a protein. The mRNA is preserved in the process, so we're not turning it into anything. We can transport it to different parts of the cell, so this is pretty big in neurons, which its nucleus is up here in the soma part, and then it has to transport it all the way down the axon to the end of the cell, so that you know, the things going on at the junction, at the synapse, can happen. But other cells can localize proteins as well. It's just kind of, in neurons, it's the most stark sort of localization. So mRNAs can be brought to different parts of the cell where different things are happening. So you can transport the mRNA along. You can degrade it in several different ways through exosomes. You can degrade it by sort of degrading the poly-A tail, the polyadenylated tail. And yeah, these are different ways that you can regulate gene expression. Because if you have mRNAs and then suddenly the cell gets a signal that says we don't need this gene on anymore, then it will signal to these enzymes like this here, or this exosome, to start degrading this mRNA, and usually that will be signaled by a protein binding to one of these untranslated regions, and then that will sort of recruit something like this exosome to come down and start degrading the mRNA. We have here too, we see there's this thing that's it's actually called an miRNA. That's a microRNA. And so there are parts of the genome that code for these microRNAs, and these can come and bind onto RNAs, and without degrading them, that can prevent them from being translated. And in fact, in experiments in the lab, that's often what you do if you want to. So there's something called a gene knockout, should be a K, knockout, which would just be removing the gene from the genome. So that would just be, if we have our gene here, we would just take this part right out and just let this form together. but we can also have a knockdown, which is adding microRNAs that are specific. Well, hopefully it's specific. You want it to be specific for a particular mRNA code that codes for a particular protein. And so if you want to see what happens to a cell if it does not have if it's unable to express a particular gene, you can go the knockout route, which is that has its own challenges. So quite often what you do is a knockdown, and that is just preventing an mRNA from being translated into protein. One of the other things that can happen with RNA is that you create these things called tRNA, which is what we're showing here. So tRNA, that's translation RNA. And what you see here is this is the actual 3D structure of it. We often portray it in this way. And you can see, like I was talking about earlier, our RNA is folding in on itself and creating these base pairs going on here and creating a shape that looks like this. And what we have on these tRNAs is we have an amino acid that will be bound to the 3' end of it. And so these are what carry the amino acids around that are ready to be incorporated into a protein. You see down here we have the anticodon, and that will just be a 3-letter, well, a 3 base pair thing. Oh yeah, I forgot to mention this too. When we're transcribing DNA, for instance, we have here in the major groove. So I talked about before that the major groove is sort of where the action happens, and that's because proteins can come in and sort of probe their amino acid residues into the major groove, and it can read this series, this sort of code of different hydrogen bonds. So we see that red means here, and here we have hydrogen bond acceptors. So if we have an H, that will hydrogen bond with these. And we see that it makes this particular order that's easy to read in the major groove. And so that's how things like the RNA polymerase actually can read, or that's how something looking around on the DNA can actually find out where a certain sequence actually is by probing inside of that major groove. Anyway, we were talking about the tRNA. So the tRNA will have so the tRNA we have here, it has the anticodon here, which can base pair with three of these bases on an mRNA. And so when it can base pair with these, it basically says, all right, this is correct. This is the correct place to put this amino acid that we have on the 3' end. And you can see what's called the genetic code here. Genetic code. We have our three base our codons here. So the 3 base things are called codons, and we can go by the first letter, the second letter, and the third letter. And we see that if we find these 3 letters, these 3 bases, then it will the tRNA that has a phenylalanine attached to it that will be the correct amino acid to add to our extending protein. And you can see we have all the other ones. We have serine and tyrosine, cysteine, arginine, uh, aspartate, glutamate, so on. So all of our amino acids are accounted for here, and in fact, there are a lot of these redundancies in it. So proline has three different codes that it can be, and you'll also notice that it's usually the third letter in it that is degenerate, as they call it. That can be kind of any one of these. So the third letter on here is often the one that it doesn't base pair as strongly, and so you can have quite a bit of variation quite often in that third letter. And I'm not going to go too far into it, but this is how you can also discern sort of evolutionary pathways, because you know that these are that codon, that the third letter in a codon is the one that's going to be able to mutate the fastest, because there isn't as much, it's not as stringent on the third letters on these. And so you can see quite often an ASP to a GLU mutation is going to be fairly common because of that third letter there. And so we see in these third letters, those are going to be the more common mutations. And yeah, I'm not going to go too deep into that. That's a whole subject on its own. So here, what we're looking at is the ribosome, and so this is the ribosome of bacteria. This is the ribosome of eukaryotes, which are what plants and animals and fungus are. And so you can see ours is a bit bigger than the bacterial ribosome, but the activity is still pretty much the same in both of them. So we see here these are our tRNAs that come into our ribosome, and so we have the different sites. There are 3 different sites in the ribosome, and so this one is the I think it's called the E site. I don't know if I have that. Yeah, so E, P, and A. So the E site is the one where this amino acid kind of gets ejected out of the ribosome. The P site is the one where I think that other picture was better. Yeah, so the A site is where you're entering, and then you go down here, and then this end of the N-terminal end, so the nitrogen on there will bond with the carboxylic acid on the amino acid from this tRNA, and then that elongating protein will end up on the tRNA in this A site. Then using energy, so we see here GTP rather than ATP. This is an instance where GTP is used. It sort of pulls the RNA through here. This one is ejected. The elongating protein attached to this tRNA is moved to the P site, and the A site is opened back up, and then we can move another one of another tRNA with a amino acid attached to it into it. Up here I have the peptide bond formation mechanism here. So we see that our protein attached, so our protein attached to the P site here is still on a tRNA. We have our nitrogen on the amino acid here attacks the carboxylic acid, and then that bonds to the elongating protein. And then over here we see that we released this tRNA that was the one we were connected to on there, and then that gets ejected out of our E site, and we now have this other amino acid on our extending protein. And so I think before I might have made a mistake. I think I called this the N terminal right here. This is actually the C terminal. Its proteins are made from N terminal to C terminal because the N on this. So we have our amino acid here. This is a really bad image of it, but this is our amino acid, and then we have the protein going off to here, and so we are extending the C terminus like that. And so yeah, I think I called that the N terminus before, but it's actually the C terminus. Just like with transcription, translation has a lot of different sort of steps to initiate it. I don't want to go too far into it, but we do have these different proteins with the methionine tRNA. If we look back up here, we see that methionine is in green because methionine is always the first amino acid. It's the start amino acid in a protein. And so we bind that onto this complex here, and we bind this complex here onto our to the 40S, the smaller subunit of our ribosome, and that creates this giant complex. Then we see that our mRNA comes in here. It causes a bunch of these proteins to attach as well, and we see our mRNA is in this site on our 40S subunit. The S just being a measure of it's called the sedimentation. It's how far the ribosome is pushed down to the bottom when you centrifuge. So larger is going to be further down your sample in the centrifuge, and that's why you end up getting this weird thing where our 60S and our 40S can come together, and it doesn't add up to 100S. I think it's 80S, and so that weight unit is kind of strange. Yeah, 80S is made out of the 60 and 40S. That doesn't add up, but these are just sedimentation units. Anyway, so we end up with this, and what happens on this is bound to our mRNA, and it scans along the nRNA until it gets to this AUG, which, as we saw up here, AUG is our methionine. So it's looking for that methionine codon. It'll process along the mRNA, and then once it gets to that AUG, then you can form the base pairing with the tRNA for methionine, and so that will recruit the 60S subunit of the ribosome and release this other stuff here. And we see here that even this process uses GTP to GDP, so that's it's an energetic process. It requires energy just to initiate this this translation here, and then so once we have this on here, then we can start doing our translation. and extending our protein. And as that protein extends, it will start folding into its native structure. But that's not quite the end of it. We're almost there. And so we can have this process called post-translational modifications. I'll just write mod. So post-translational modifications are things that can happen to your protein after it has been translated. And so things like methylation, acetylation, sumolylation, those are things that mostly happen to the nucleosomes, which we talked about down here. So those are the epigenetic regulation of nucleosomes. So things like phosphorylation and dephosphorylation, for that matter, and glycosylation are not uncommon. Ubiquitination, so ubiquitin and the sumo protein, I forget its exact name. These are both proteins that they're little small proteins that you can actually attach to your larger protein. And I'm not as familiar with sumolylation. It's been a while since I looked at that. But ubiquitination, what ends up happening is you start adding a bunch of ubiquitin molecules on it, and this will sort of mark a protein for degradation. And so this will be a protein that's sort of marked as we don't need this protein anymore. We've flagged it to get rid of it. Proteolysis is another fairly common post-translational modification. That's basically just you could have a protein that it's one protein, but it's in these two domains. And you could have another protein, some kind of enzyme, come down and actually cut those domains apart from each other. And what that can do is it can open up maybe an active face of the protein there, or things like that. So that's a way that you can, you can have your protein full like this, and maybe it's not doing anything. Then you have some enzyme that is signaled to activate, and that'll come down and cleave these proteins from each other, and that will cause them to become active. This process, so I'm recording this in 2020. This process actually occurs in the spike protein of the coronavirus. And so the spike protein is one that is cleaved. I believe it is cleaved by a protein in our own lungs. So it's using our protein to cleave its spike protein, which then is a membrane-bound protein, and that opens up that spike protein. It opens up faces that allow it to get into our lung cells. And so that's sort of a topical example of this proteolysis happening. And so let's talk a little bit about glycosylation. So I've talked about phosphorylation before, and that was, it's just adding that phosphate group onto usually a serine, a threonine, or a tyrosine, the hydroxyl groups that those contain. And so you add this on here. This has this highly negative charge, which can cause conformational changes in your protein. So it can either activate or deactivate your protein in different ways. And so that's a way that the activity of proteins are controlled in the body. We can also have, so this is glycosylation. And so we can This is our protein, and this is pretty common for proteins on the surface of a cell. So this is outside the cell out here, and you can add all these different chains of sugars onto the proteins. Uh, this happens on proteins inside of cells as well, but the sort of the canonical case is here on the outside of cells, and you can so this shows that you can bind these sugars, so we can have fructose and galactose and mannose and things like that. And they can bind onto nitrogens, and so that would be in yeah, so it's binding onto a nitrogen on a side chain, or it can bind onto a serine or threonine side chain uh, on your protein, and you can have these different O glycans and N glycans. I'm not going to get too much into that. And this is just an image of so this is a protein structure, and this is the protein structure with this giant uh, sugar chain on it. So this is a heavily glycosylated protein. And so those are just the different kinds of post-translational modifications that can happen after translation. I believe that is everything I wanted to say in this video. So that is just a little bit about nucleotides, a little bit about transcription, translation, post-translation modifications, and that is like I said, what we call the central dogma. And so this playlist is a nutrition playlist, but and this maybe doesn't seem like it has that much to do with nutrition. I mean, we do eat DNA when we eat anything. It's made out of cells and it contains DNA, but we don't really get much nutrition value from DNA or from these nucleotides. But a lot of metabolism and nutrition and things like that has to do with regulating the transcription and translation of genes, as I talked about before. I forget where that was, but I talked about how I think it was over here. Oops. So over here, yeah. So insulin. If you're into nutrition, you probably know a little something about insulin. It's what regulates blood sugar, and so we see that insulin has an effect on this transcription, and that's why transcription. So if we're going to talk about carbs, about sugars and carbs, then we're going to talk about insulin. If we're going to talk about insulin, we're going to talk about gene expression. If we're going to talk about gene expression, we're going to talk about transcription. And so that's why I went over all this, even though it on the surface of it doesn't seem like it has that much to do with nutrition and metabolism. But I think that is everything I wanted to say in this video, so I will see you in the next one.